In this video, I want to tell you a little bit about DNA sequencing, since the rest of this module will be how we are going to assemble, annotate, analyze a whole genome assembly. So DNA sequencing has a relatively long history. The first method for doing DNA sequencing is called Sanger sequencing or dideoxy chain termination sequencing. This first was available in 1977. It was created by a scientist named Fred Sanger, who ended up winning a Nobel Prize for it. And it takes advantage of the chemistry of the bases in terms of the 3' OH. So dideoxy means that there are some bases that lack the OH at that 3' location. They instead have an H, and so you get a termination of the chain. And we'll see a video about that in just a couple of minutes. You can see some of the different advances in sequencing. The first mitochondrial genome, the Human Genome Project, first cellular genome, which was a bacterium, the complete eukaryotic genome, the first completion of the Human Genome Project, and then the invention of second-generation sequencing, which we'll talk more about a little bit later, the Human Microbiome Project, third-generation sequencing, and all of these different advances. So what we're going to do through this video is look at the different kinds of sequencing technology and how they work.
Oh, hey, let's go back to the basics and explore the technology platform that has been regarded as the gold standard for many years. Yeah, you guessed it. I'm talking about Sanger sequencing by capillary electrophoresis. Many might ask, why is it called Sanger sequencing? Well, Sanger sequencing is named after the inventor of this groundbreaking technology, Dr. Frederick Sanger, who I like to call Fred, who developed this method over 40 years ago in the mid-70s. So what are the basics of Sanger sequencing? Let's take a look at our lab book. It all starts by having a short primer binding next to the region of interest. In the presence of the 4 nucleotides, the polymerase will extend the primer by adding on the complementary nucleotide from the template DNA strand. To find the exact composition of the DNA sequence, we need to bring this reaction to a defined stop that allows us to identify the base at the very end of this particular DNA fragment. Sanger did this by removing an oxygen atom from the ribonucleotide. Such a nucleotide is called a dideoxide nucleotide. This is analogous of throwing a wrench into a gear. The polymerase enzyme can no longer add normal nucleotides into this DNA chain. The extension is stopped, and we now need to identify what it is. We identify the chain terminating nucleotide by a specific fluorescent dye, 4 specific colors to be exact. Sanger sequencing results in the formation of extension products of various lengths terminated with dideoxide nucleotides at the 3' end. The extension products are then separated by capillary electrophoresis, or CE. The molecules are injected by an electrical current into a long glass capillary filled with a gel polymer. And during CE, an electrical field is applied so that the negatively charged DNA fragments move toward the positive electrode. The speed at which the DNA fragment migrates through the medium is inversely proportional to its molecular weight. This process can separate the extension products by size at a resolution of 1 base. A laser then excites the dye-labeled DNA fragments as they pass through a tiny window at the end of the capillary. And the excited dye emits a light at a characteristic wavelength that is detected by a light sensor. Software can then interpret the detected signal and translate it into a base call. When the sequencing reaction is performed, in the presence of all 4 terminated nucleotides, you eventually get a pool of DNA fragments that are measured and separated base by base. What you will get in the end is a data file showing the sequence of the DNA in a colorful electropharogram, and a text file which you can use to answer the questions you may be asking. And that, in a nutshell, is Sanger sequencing. For more information, download the free DNA sequencing guide through the link below. I hope this video was helpful on the basics of Sanger sequencing, and I'm sure you'll have more questions. So submit your questions at thermofisher.com/ask. and subscribe to our channel to see more videos like this. And remember, when in doubt, just seek it out. Oh, hey.
So that type of sequencing analysis is how scientists started doing the first genome sequencing. And again, the first organism that was actually sequenced, like the first cellular organism, was a bacterium. And they had tons of these machines that would run the sequences all at one time. Now, some of the challenge here is this isn't very high throughput. You can only sequence one fragment at a time, so it can be very, very slow. And we also didn't really have a lot of the computational power to assemble and put all of these sequences together. So to sequence, for example, the first bacterial genome took something like 3 months of these machines running 24/7. Now, in the mid-2000s, and this is why I always laugh about biology and how biology is such a changing field, this type of sequencing technology, next generation sequencing, wasn't even introduced until 2004, 2005. So this was something that we didn't even talk about when I was an undergraduate student. It didn't exist. So this technology, once it existed, is high throughput sequencing or shotgun sequencing, because you take a whole genome, you fragment it into a bunch of small pieces, and you start to sequence it. There were a handful of different companies that were trying to do this. The 3 are right here. And really the winner ended up being Illumina, and we still do Illumina sequencing today. So we can call this either next generation sequencing, or sometimes we call it second generation sequencing. And there were a lot of benefits to this over first gen sequencing. For example, the preparatory steps are fewer. You don't have to run the PCR first. You don't have to purify all of this DNA and run it out on a gel, so it gets to be a lot faster, right? No electrophoresis. The other thing is that depending on how complex the run is, you could sequence a whole genome on one machine in a week or 2, rather than having to sequence a whole genome on many, many, many machines over weeks and weeks and weeks and weeks and weeks. And again, Illumina has really been the company that has been the most successful in this area, and they kind of dominate the field. There are a lot of different machines that they have on the market, and it really depends on your applications for the machine, which one a scientist might choose. For example, if you're wanting to do something like a human genome project or a large eukaryotic genome, you're probably looking somewhere over here on the right, maybe not even the NextSeq, but maybe a HiSeq or a HiSeq X10 that can output, a terabyte worth of data. If you're on the bacterial side, maybe a mini or a MiSeq would really be sufficient for you. But one thing to notice here, max read length, 150, 300, 150, 150, 150. Sanger sequencing can do a fragment of up to approximately 1,000 base pairs, and they're usually pretty accurate. These next generation sequencers really use what we call short read sequencing, very, very small fragments. And as you might imagine, that provides a challenge when we're trying to assemble a genome, which is what we'll talk about on Thursday. So how do we prepare DNA to be sequenced using Illumina technology? Well, broadly, what they do is fragment the DNA. That's why they call it shotgun sequencing. They put it into thousands and thousands and thousands of little pieces. And then during the library preparation step, adapters are glued or ligated to the ends of those fragments. And those adapters allow the fragments of DNA to be in a flow cell, which is part of the machine. And then there's a PCR reaction that occurs in there, much like what we see with Sanger sequencing, but we can do millions at a time. So that's why we call it high throughput sequencing. And what I'm going to have you look at next is a video put out by Illumina on how this technology works.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated, similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities of next-generation sequencing.
So again, Illumina sequencing is really the most common and most popular type of sequencing that we do. And all of the sequences that we'll be looking at in class are derived from Illumina. Now, when you get the file back, the files are what are called FASTQ files. They're not FASTA files like we've been dealing with before. So every FASTQ file has 4 lines. Now, sometimes, depending on how big the window is that you open these to look at, like the sequence might wrap around onto multiple lines, and then this line down here might look like it's on multiple lines. But if you really spread it out as far as you could, you would have a line that starts with this @ symbol, and that represents the ID. So it'll tell you something about the fact that it's this read, it came from which machine. You don't need to know this information, it's just the information that comes off with the machine. Line 2 is important information. This is the actual sequence that the machine read. So it read this order of bases. Line 3 is just a plus sign, it's a separator, so you'll always know, even if you do see the sequence extend onto a second or third line, technically line 3 is going to be this plus sign. And then you'll see more letters, but not DNA-based letters, just regular letters. And these represent the quality scores. And I'll tell you on the next slide how we determine the quality scores, but theoretically you just line up each base, right, with the letter in the line below, and that is the quality score. When we're looking at the quality score, what we're looking at is how the sequencer has estimated its own error rate. And remember, we're talking about how it's looking for fluorescent signals. And so if there's ever an error, you would think that for the most part, the correct base would be there, 90% of the time, 95% of the time, 99% of the time. So that signal will be the brightest because that base is the one that's there. But let's say, that base isn't the brightest, or there's not one that's really brighter than another, or maybe it's 51% versus 49%. So in that case, the sequencer makes, I don't want to call it a judgment call, but that's kind of what it is. It kind of makes a judgment call. It decides which base is going to be the one it puts in the sequence, and then it gives it a score. But that score is going to be a lower score if there's less confidence in that being the correct base. So the error is converted to what we call a Phred score, and how that works is not terribly important for you. But a Q of 20, so a Phred score of 20 means there's about a 1% chance of an error. It involves the probability of that base being an error. Q30 is a 0.1%, Q40 is a 0.01%. So the higher the number here on the Phred score, the lower probability that there's an error in the sequencing read. So then those scores are converted to letters. So you might think A is great, right? Because you want an A in every class, and A is the best grade. In this case, I represents a really high quality read. F is maybe a more moderate probability. A is high error, low quality. So if we look here, we see A, B, F, G, F, H, G, H, F, right, H. And so what we tend to see, if you really were examining these scores, is that the lower quality reads are usually at the very beginning and very end of each sequence. So everything right at the very beginning and at the very end will sometimes have lower quality than what's in the middle. So we have more confidence in the middle. And that'll be really important when we think about processing our reads and looking at the quality of our reads. So one of the other things that was talked about in that Illumina video was doing paired end. We read from both ends. This usually improves the assembly because you have more coverage, and it helps to span maybe repeated regions, And in RNA sequencing, it can even help to provide things like orientation. So when we start to talk more about things like RNA sequencing, we'll talk more about how you can determine the orientation of the reads. So that's second generation, next generation, high throughput sequencing. And then, we can briefly talk about the more modern type of sequencing, third generation sequencing, which is also sometimes called long read sequencing. Now, remember, Sanger sequencing has a benefit of, it's up to 1,000 base pairs. And then, when you get to the short read sequencing, maybe 150 to 300. So you have the pro of Sanger sequencing of a longer read. You have the con of short read sequencing of shorter reads. But with long read sequencing, with the Sanger sequencing, it takes a long time to get that many bases and to assemble a whole genome. With the short read, you can do the whole genome on one run. So pros and cons. This long read sequencing, this third generation, is really trying to address the pros and cons of the first two generations, right? Why would you come up with a new technology if it wasn't going to be an improvement? So some groups report that they can get a single fragment, not 1,000, not 150 or 200 base pairs, but literally up to 2 million base pairs. More typically, what you would see is anywhere from 10,000 to 100,000 base pairs in one read, which is orders of magnitude higher than what you would get even from Sanger sequencing. So often what you're looking at is with short read sequencing and with Sanger sequencing, you're amplifying and then sequencing. With long read sequencing, in some ways, you don't even have to amplify, depending on the type of technology. You literally put the DNA in and it starts to read it without any sort of processing. What short read sequencing does is it uses the computer to sequence a large fragment. You're not actually sequencing a bacterial genome. You're sequencing 150 to 300 base pairs and then gluing them together later. And on Thursday, when we talk a little bit more about assembly, I'll give you an analogy that hopefully will make some sense with that. So when you're talking about long read sequencing, some of the advantages are that it absolutely is really, really helpful with genome assembly. Because if you have to put millions of tiny pieces together, that's much harder than putting together a few really large pieces. It can help you identify variants because you can see where maybe there's a mutation where that could get lost in the signaling in the short read sequencing. One of the long read sequencers is highly portable. But again, because you're not doing the amplification and sequencing, you're just doing the sequencing, you can start to get answers and data in real time. And depending on which long read technology you're looking at, some of these have been adapted to allow you to even do direct sequencing of RNA. And when we talk about transcriptome analysis, we'll talk more about why that would be such a benefit. But as of right now, with the exception of Nanopore technology, when I say that we do RNA sequencing, we don't actually sequence RNA. We take RNA, we convert it to DNA, and then we sequence the DNA. But some of this technology, this new third generation technology, allows us to directly sequence the RNA itself. Some of these long read sequencers can also allow us to do things like look at epigenetic modifications. Where is there methylation on the DNA? And you lose that with the short read sequencing and with the Sanger sequencing through that PCR process. You lose whatever epigenetic modifications might have been there. There are two major technologies. One is by PacBio. It's called SMRT, Single Molecule Real-Time Sequencing. The other is a company called Oxford Nanopore, and their sequencer is called the Minion. It looks like it should be Minion, so that's why I like the Minion. And I have one of these that one of my friends is borrowing right now, and his name is Kevin, as it would be, of course. So what I have next are just a couple of videos for how these technologies work, and then we'll wrap up these slides.
PacBio Hi-Fi sequencing technology gives researchers a comprehensive, multi-omic view of the genome, including GC-rich regions and long repetitive sequences that other technologies can't fully resolve. Here's how HiFi works. HiFi sequencing is built on single-molecule, real-time technology. Its accuracy comes from repeatedly observing the same native DNA molecule as the polymerase replicates it. First, DNA from nearly any organism is converted into a circular template by ligating adapters to both ends without PCR amplification. This avoids bias in highly repetitive or GC-rich regions and allows for the simultaneous detection of genetic variants and methylation status in a single assay. Next, primer and polymerase are added to the library, and the sample is ready for sequencing. Sequencing happens on a SMRT cell, which contains millions of tiny wells. Inside each well, the polymerase gets to work, building a new DNA strand base by base from the native template. As each base, A, C, G, and T, is added, a unique pulse of light is emitted and recorded in real time. These pulses create a continuous record of the molecule as it's read again and again. HiFi also captures epigenetic information automatically. When a base is modified or methylated, subtle shifts in the kinetic signature of the polymerase's incorporation rate reveal rich epigenetic insights like 5-MC and 6-MA methylation. This happens automatically on every sequencing run without additional library prep. HiFi reads are generated by combining information from multiple passes around a single DNA molecule. The system combines these repeated observations to produce long, highly precise reads, exceeding 99.9% accuracy. Powerful on-instrument algorithms and analysis tools turn this raw data into rich, multi-omic insights, revealing the genome, including complex structural variants and dark regions, the transcriptome with full-length isoforms, fusions, and splicing, and the epigenome with base-level methylation patterns captured natively. All in one run, all from the same molecule. With HiFi long reads, researchers can do everything from interrogating the hardest regions in inherited disorders to powering population-scale precision medicine with complete, accurate genomic and epigenomic data. HiFi sequencing removes blind spots and delivers the accuracy needed to understand biology at every level.
Okay, so that's PacBio, and then we can look at Nanopore sequencing.
Now, the third type of NGS sequencing that we use is nanopore sequencing, which is also known as Oxford nanopore sequencing. The DNA is passed through a tiny protein pore, which is known as nanopore. Now, this nanopore sequencing is completely different. It's out of the league. It's on its own, right? So we have a protein channel, right? A pore is there, just like a protein channel, which is known as nanopore. And the DNA is passed through that nanopore. While the DNA is passing through the nanopore, the double-stranded DNA cannot pass. So DNA strands need to be separated. Only single-stranded DNA is inserted through that nanopore, right? And each base alters the electrical current in a unique way, right? Because we put current for the movement of the DNA, because the DNA backbone is negatively charged, so we can assume that the DNA will move towards a positive charge, right? So if we distribute the current as a negative to positive, then the DNA will flow from negative towards the positive end. As it's moving towards the positive end, the strands are segregated, single-stranded DNA is migrating. And as the DNA is moving, the bases alter the electrical current in a unique way. And we have a detector out there, the device that reads the signal in real time. The way the bases are changing the current signal is measured. The longest reads length are possible. It can go over 1 million bases. So this is a technology that can sequence largest reads. Largest read means here 1 million base. Over 1 million base can be read at a single go with the help of nanopore sequencing technology. That is the advantage of nanopore here compared to the other Illumina and ion torrent sequencing. Slightly lower accuracy, but improvement is going on very fast to make it better over time. It is used for whole genome sequencing because the whole genome size is much bigger. So we need to read lengthier form of the DNA. For structural variations and also for pathogen detection, we use Nanopore sequencing or Oxford Nanopore sequencing. Here you can see one illustration of the process. So the current is moving, negative charge outside, positive charge inside. We have the protein nanopore central channel and the DNA. As I told you, the DNA strands are getting separated on only a single-stranded DNA, which has a large negative charge in the backbone, is moving towards a positive charged end. And as it is moving in this current, these bases that are present here are going to alter the current here inside of the membrane, because inside we have positively charged ions here, or positive charge in terms of the current. But the bases, the more bases coming in, it's going to change the positive charge towards the negative value, because it will be neutralizing the charge that is out there. So this change of the current intensity as the bases pass by, the way A changes the signal is not same as the G will change, as not same as the C will change or the T will change. For all the different bases, the change will be different. The current intensity will be different. So with time, if we measure the current intensity, we are going to find out a pattern. And we know how and at what intensity A will change, and what intensity C will change, at what intensity T will change, at what intensity G will change the current intensity. And by knowing that, we can easily determine the sequence of the DNA fragment that we're analyzing with Nanopore. And in this case, we don't need to break it apart, because it's a continuous process. Through the pore, the single-stranded DNA continue to migrate until and unless if there is any mechanical shearing, then fragments of the DNA can be lost, but that's a negative side. Without that, stretch of a lengthier DNA can be transferred through the nanopore. Okay, that can be done in this case of nanopore sequencing. That's nanopore sequencing for you. The best uniqueness here is it can go over 1 million bases as a read.
And so again, as both of those videos talked about, these two types of sequencing technology are great because they do allow us to look at long reads, look at variants, look at modifications. So you might ask, well, didn't you tell us a little bit ago that Illumina is kind of like the gold standard for sequencing, is what everybody does and blah, blah, blah. It is. But not because it's necessarily better, but it is a lot cheaper. Doing Nanopore and even PacBio sequencing are still relatively expensive and you know, a little bit out of reach still for a lot of researchers, myself included. We can only do PacBio, and we only do PacBio when we're looking at eukaryotic genomes. And again, it's not cheap. So we do it because we want to, and we want to have that quality, and we need it to get as continuous as possible a eukaryotic genome. But it is out of reach for a lot of people. And of course, you know, that read length itself, it really matters. So short reads, you know, anywhere from 100 to 300 base pairs are very, very accurate. But of course, you do run into these long repetitive regions, especially in eukaryotic genomes, where it's nearly impossible to assemble the full genome because you have these repetitive regions and you don't have any sequence that spans the whole region. So with long read sequencing, you often can sequence through an entire repetitive region, so it's much easier to generate a continuous chromosome, for example. Now, I found the most updated PacBio video I could, and the Nanopore one, the company itself hasn't put out a video in a while, so that's why I chose the Nanopore video narrated by that other gentleman. The accuracy is getting better every time. You saw PacBio has very, very high accuracy. My understanding of Nanopore is that it's still a little bit lower than short read, but at the end of the day, you know, you also have those really long reads. So it depends on your application. It depends on your funding. Now, if I had, you know, better funding resources, you know, if I had thousands of dollars to drop every time I wanted to do sequencing, I would 100% always do long read sequencing. And I used to say that I would only do Nanopore because I think Kevin is so cute, but I've been converted because of Jair. Those of you who know Jair, I've been converted to PacBio. So if I could afford it, I would do everything on PacBio. I can't afford it, so that's why I still do a lot of Illumina. And of course, when I do microbiome analysis, Illumina is fantastic. I don't need more than a couple hundred base pairs anyway, so why would I spend the money to do something more? Now, one of the other things that we think about when we think about sequencing is how much coverage are we going to get? And by coverage, it basically means on average, how many times was each base sequenced? And what you're looking at here is, these bases, kind of where my mouse is, each got sequenced 5 times. But these bases over here each got sequenced once. So what you have to calculate is the average coverage. So you take the number of total reads, which will come out of the sequencing machine, the read length, you multiply those together, and you divide it by the overall genome size, and that'll give you a number, a coverage number. High coverage generally means you have better assembly, because there are going to be some regions where you have like a ton, and there will be regions where you have fewer reads that cover it, but hopefully more than one. Low coverage generally will give you some fragmented contigs. And don't worry about this term yet. We'll talk more about it later. But it generally means that you probably didn't cover the whole genome well. Now, what does sequencing not give us? Well, it doesn't give us an ordered chromosome. It just doesn't. We'll talk more about how we can try to get there when we talk about assembly on Thursday. Sequencing does not give us annotated genes. How could it? It just doesn't. It doesn't give us any functional interpretation. What does this genome do? All it does is give us reads. That's it. It's just a big file that looks like that screenshot I showed you earlier. Doing the assembly to try to get as close to a chromosome as possible, doing annotation so that you can determine gene function, are separate steps that we'll talk about as we continue in this module.